Friends, this is Vaidam and welcome to my YouTube channel. Today we are going to discuss the part 3 or the last lesson regarding the basis of life. So we are going to start from there. Yesterday we have discussed three major organelles of the cell, which is plasma membrane, nucleus and endoplasmic reticulum. Now we are moving further to learn the further organelles. Today we are going to start our lecture with the Golgi complex, which is the next organelle which we need to discuss. Okay, now from the yesterday point of view, this is the nucleus which contains the genetic material and which provides the information to control all the activities of cell. And in attached to this, in a further attachment to this is endoplasmic reticulum. We know that there are two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. So we know that the endoplasmic reticulum is involved in production of protein or polypeptide or lipid synthesis. But the protein and polypeptide, polypeptide, and lipids produced are not in final form. The final form is given by this very known organelle by Golgi complex, which was discovered by Camillo Golgi. So this is the Golgi apparatus. Golgi apparatus is a stack of membranes, and basically it has two phases. One is cis phase and other is trans phase. Cis phase is the phase where the vesicles which are budding from the endoplasmic reticulum are attached to the Golgi complex, and from the trans phase where the mature vesicles move from the Golgi complex to the various part of the cell which require them. Okay, now I'm going to tell you that, as we know, rough endoplasmic reticulum produces protein. Proteins is given final form in the Golgi complex, which cause modification of protein by glycosylation. Modification of protein by glycosylation. It is basically an organelle which is involved in the transport, storage, and modification of protein produced by endoplasmic reticulum, rough endoplasmic reticulum. So, as the protein is modified, it is transported where it is destinated. Maybe it might be destinated route 1, which is this one, route 1. This could be that it could be a secretory protein that is required by the extracellular matrix. So this secretory vesicle is formed from the trans phase of Golgi complex, and this is transported to the plasma membrane, where through exocytosis protein is expelled out, or the modified protein is expelled out. Now, number two, the second part, I'm going to tell you that the vesicle which may be produced contains transmembrane protein. First of all, it's a secretory vesicle. It could contain the digestive, some sort of enzyme that is required by extracellular matrix or the other cells. So that is produced by the cell. It may be secretory cell, so it has to produce that protein and is secreted out. Through the process of exocytosis, that protein is expelled from the cell. The second part is vesicle containing transmembrane protein. This is one of the very important perspective, how the cell membrane is formed, how it maintains its fluidity, how the cell size grow. This is one of the part which could answer that question. As the protein is produced by rough endoplasmic reticulum, this way you can consider protein that is embedded in the membrane of rough endoplasmic reticulum like this, and this embedded protein comes to the Golgi complex, where it is modified, maybe glycosylation attached to form a glycoprotein. This budded vesicle comes to this one and it contains glycoprotein, and finally that is attached to the plasma membrane. So the protein formed here is actually became the part of plasma membrane. So this is how the plasma membrane is enhanced. There are number of proteins which have to attach that vesicle into the plasma membrane and it form its part. So first part is as exocytosis, it is secreted out. Second route, we can say that the vesicle contains transmembrane proteins. Yesterday we have discussed in the plasma membrane structure that transmembrane proteins which can act as receptors are present in the plasma membrane. This is the way how the transmembrane proteins are obtained in the plasma membrane. Okay, part third. Third part. In the third part, I am going to discuss that the Golgi complex may be involved in the formation of another organelle known as lysosome. So, Golgi complex is involved in the storage, transportation, and modification of proteins to the destined organelle of the cell. So, lysosome is here. We know that the budding phase, the mature phase, releases the organelle that is called lysosome. Lysosome contains the digestive enzymes of the cell. It will help the cell when it want to autolyze and when it want to be autolyzed, when it wants to be destructed. So autophagy is the mechanism which is performed by lysosome, as it contains digestive enzyme in the lysosome. Okay, so further moving on, when a certain food particle that enters the cell, or antigen, or a phagos antigen, or some sort of foreign particle enters the cell like this, it has entered the cell, foreign particle, it fused with lysosome, or it may be food particle, and form phagosome. So lysosome provides enzymes for the digestion of that particle or food particle, or it may be foreign invader. So this is how the lysosome functions, and it forms phagosome. But lysosome can perform the function of autophagy. The next point I'm going to discuss that there are a variety of lysosomes like microbodies in the cell. This is one of the microbodies in the cell that may be that I have written yesterday, that may be peroxisomes, glyoxysomes. These are variants of these could be considered as variants of lysosome, and they perform also the digestive process for the cell. Peroxisomes is one of the organelles that is present in all type of eukaryotic cell. Eukaryotic cell that are not bacterial cell in a simple sense. So peroxisomes contain all the digestive enzymes, and they are present in plant, animal, and everywhere. And the lysosomes are those organelles which are only found in plant cells and fungi cells. So they are mostly present in when seeds are germinating, and peroxisomes are present in all the cells. So they are involved in basically they are breakdown of fatty acids. So peroxisome and lysosome are these organelles. So this is one of the basically structure how the Golgi complex plays an important role. It stores, it transport, and it modify the proteins which are produced by endoplasmic reticulum, and then they are further destined to the organelle which they or the route they want to. So I have discussed route 1, route 2, and route 3. So these are the roles played by Golgi complex. Next, moving to the next organelle, that is cytoskeleton. I am going to discuss cytoskeleton a bit of here. These, as you know, these organelles are moving, so they require certain pathway. What pathway they are required? I'm going to discuss that in detail. So, how these vesicles move through the cell? So there might be some sort of skeleton. Everywhere there requires a path or route for that. So that is cytoskeleton. Cytoskeleton is the next organ we are going to discuss. Okay. As we know, this cyto means cell, and skeleton means the structure which supports some of the organism in which they are present. So cytoskeleton is actually the skeleton of the cell. So it basically gives the shape and form to the cell. Shape and form to the cell. So it maintains the integrity of the cell. How the cell is destined to be shaped is done by the cytoskeleton. We know that cytoskeleton comprises of three types of filaments. So these are microfilaments, tubules, and the third one is intermediate filaments. So cytoskeleton is composed of three types of filaments: macrofilaments, tubules, and intermediate filaments. They do have different role, but they form the skeleton of the cell. Microfilaments are thin thread-like structure, and they are basically made up of actin molecule, actin protein, which are also attached to different other proteins to form the structure of the cell. So basically, they provide the root pathway through which the organelles or the vesicles move. So microfilaments are the structures which are spread out in the cell or in a cortex or in an area where the traffic, Golgi apparatus traffic is moving on. Next is tubules, and basically microfilaments are abundant in muscle cells where there is a construction of muscle. So actin is associated to another protein which is called myosin, and these two proteins are involved in the contraction of muscles. We will be discussing in detail how the actin and myosin are involved in the contraction of muscles in the human physiology when we are going to survey the part of human physiology. The second part is tubules. Tubules basically are made up of protein called tubulin. So tubulin is also a dimer protein. There are two particles. There are two particles that are associated, or two molecules associated in that shape that they are attached together, and they form the filament. When it gives the polarity, tubulin molecules are attached in such a way that it gives the polarity in such a way. Tubulin molecules are attached in such a way that it gives polarity. One end is negative, the other end is positive. Tubulins are attached in that format. Dimers. So I have drawn a journal structure here. From the negative end, dimers always get removed, and from here tubulin molecule is being attached. This is how the shape of the tubulin is attached. How they are formed? Two tubulin molecules take up this one strand, and one strand, two tubulin molecules with many other molecules polymerizes to form a strand, and that 13 strands combine together to form a tubulin tubule structure. So this is how the tubulin act, and it play a important role in the formation of, basically it forms a microtubule. This is how basically microtubule is formed. As you know, the microtubule is a structure which is involved in the movement of chromosomes during cell division. So this is how the microtubule are formed. I have discussed in the previous introductory part that near the nucleus we have an organelle in animal cell that is known as centriole. So the centriole is in the area centrosome from where microtubule emerge. They are in a right angle triangle format. So they are perpendicular to each other from where tubulin or microtubules, either tubules and microtubules emerge and involved in the movement of chromosomes during the cell division. The third part is basically intermediate filaments. These are filaments are in between the microtubule and tubules. These are present in the basically periphery of the cells and form various junctions. So it is how they help in the attachment of one cell to another cell. That is the role played by intermediate filaments. They are also thread-like structures, but they have the size intermediate to macrotubule and tubules. Tubules form tubulin and dimer, and that results in the formation of tubulin assembly results in the formation of strand. That 13 strands assembled together to form macrotubule. A macrotubule play a role in the cell division. This is how I am going to discuss briefly the cytoskeleton. Cytoskeleton is the skeleton of the cell. So moving towards another organelle, which I am going to discuss here, is about mitochondria, which is also known as the powerhouse of the cell. So this is one of the organelles that plays an important role, so without which we are unable to survive even, and the problem in that cause very severe diseases. So mitochondria is one of the important perspective I am going to discuss here. Mitochondria is present in every eukaryotic cell, so it plays a vital role. Except, I am going to mention here that mitochondria are not present at not present are not donated by father when there is a fertilization of sperm and egg, but mitochondria basically is what are which provides energy to us. And the structure of I'm going to discuss the structure of mitochondria. Mitochondria is double membranes organelle. The outer membrane is smooth. The inner membrane has foldings. This is how it is easy to identify mitochondria in the cell. So this form finger-like projections called crystal, and inner area is also called matrix, which contains basic fluid of the mitochondria. This is here is the site where chemical reactions occur. Maybe it might be at the crystal, maybe it might be in the matrix. Okay, that is the outer membrane, and it's the inner membrane, and it's the inner membrane of the cell. So this is structure of mitochondria, and it is also known as the powerhouse of the cell. Mitochondria comes in a variety of forms and shapes, and it has very degree. It may be rod-like, it may be spherical. So mitochondria is one of the organelles where the Krebs cycle occurs, and it provides energy for the functioning of the cell. So that is the reason it is known as powerhouse of the cell, because it contains all the key enzymes that are required to break the molecule to the energy yielding molecule, which is also known as ATP, which is also a it's a type of nucleotide, adenosine triphosphate. It is also known as energy currency molecule. So it's a nucleotide, and as the energy is released, which is used by the cells, it converts into ATP, converts into ADP with the release of phosphate. So basically, mitochondria is an organelle which is present in all the eukaryotic cell and provide energy to the organism. So this is all about the mitochondria. Now we have discussed about all the organelles: nucleus, plasma membrane, lysosomes, endoplasmic reticulum, Golgi apparatus, and mitochondria. That how they are important in the functioning of cell. So I am going to end my lecture here, and this we have discussed even the cytoskeleton in detail, and I am going to tell you briefly about the ribosome. Ribosome has two subunits, and it is involved in the proteins. Previously I told that it is the organelle that is involved in the proteins, but it's a subparticle. So you can find questions regarding the basis of life in my page, on my page, which is known as Incredible Waha. So you can find the questions regarding the basis of life, which would be posted after this lecture. And if you like the video, please like, do comment, and subscribe.